FluoRender tutorials. 4D Tracking and Analysis. Part 1. Selective Tracking. You can use FluoRender's paintbrush tools to select and track cells manually. Click Paintbrush from the main toolbar to open Paintbrush setting window. Paint and select several cells to determine the paintbrush settings for easy selections. This particular dataset has two layers in the Z direction. Cells are in the top layer. We can use the clipping planes to cut in the Z direction, so that the background signals are not selected. Before we start tracking a cell, we need to enable a 4D script. The 4D script, selection_tracking, can be found in FluoRender's installation directory, in the Scripts folder. Let's copy it to the data directory for easier loading. Go back to FluoRender, and in the 4D script tab of the Record/Export panel, check Enable 4D script. Click Browse, and in the Open File dialog window, select the script file we just copied. Open the tracking window from the main toolbar. Go to the Linkage tab and enable AutoLink. The AutoLink function links a selection at one time point to the selection from its previous time point. Go to the Modify panel and enable AutoAssign. The AutoAssign function assigns an ID to the selection after each time you paint in the render view. Starting from the first time point, paint and select just one cell. Move to the next time point and select the same cell. You can use the keyboard shortcut D to move forward and S to move backward. Repeat for all subsequent time points. Notice that once AutoLink and Assign are enabled, you only need to make sure that the same cell is selected at each time point. However, you can only work on one cell at a time. Since the AutoLink and Assign functions assume that you are selecting the same cell from each time point. If the cell's signal is getting too low, you may need to adjust the paintbrush settings to select it. Lower the paintbrush threshold value so that the cell can be selected. You may also want to adjust the viewing angle and clipping plane settings as the cell moves over time. Repeat until the cell is selected from all time points. Once you reach the last time point, this one cell has also been tracked manually. Don't forget to turn off AutoLink and Assign if you would like to examine the track of the cell. Rewind to the first time point. Go to the Selection tab and type the ID of the cell and press Enter. The cell should be selected. You can play back or step over time to observe the tracking result. As long as you have selected the cell correctly from each time point, the tracking result should be perfect. We can track a second cell using the same method. First, enable AutoLink and Assign. Increase the threshold value in the paintbrush settings to the number we used to select the first cell initially. Select a neighboring cell and repeat the selection for all subsequent time points. Use the keyboard shortcut D to advance to the next point. Observe the movement of the cell carefully and make sure it is selected correctly. If no more cells are to be tracked, disable AutoLink and assign after the second cell. Rewind and examine the tracking result. We can type all in the component ID field and load both cells. Play back our step over time to examine the tracking result. To save the tracking result, go to the Track Map tab and click Save. The saved track file can be loaded later for viewing or editing. Part 2. Semi-Automatic Tracking. You can track a group of cells by taking advantage of FluoRender's component analysis features. Starting from the first time point, select a group of cells. Usually, these can be cells of importance to research. In the Component Analyzer window, check Use Selection and then click Generate to segment the selected cells. You need to save the segmentation result for the first time point. To do so, click Save Mask in the Datasets panel. We can see that 2 files are created in the Data folder. These mask files store selection and segmentation information. Advance to the next time point. The selection mask is highlighted for the previously selected cells. Clear the previous selection and select these cells again. Notice that the colors are from the previous time point, which can be used to identify the overlapped region of the same cells. Click Generate in the Component Analyzer to update the segmentation of current time point. Save the masks of the second time point. Since saving masks is an easily forgotten operation, we can use a 4D script to automate mask saving. Go to the 4D script tab and load a 4D script called save_fetch_mask. You can find this script in FluoRender's installation folder. For each time point, clear the selection first. Then, paint and select the group of cells. Segment the selected cells with the Component Analyzer. Advance to the next time point. The masks are saved by the script when you advance in time. Repeat the operation for all subsequent time points. For completing a series of operations with ease, remember these keyboard shortcuts. C for clearing selection. Shift for the selection brush. And D for advancing one time point. Also notice that you need to make gaps between selected structures for the Component Analyzer to easily segment them. You can change viewing angles or adjust clipping planes in the process of selection. Use the colored components from the previous time point to find out the overlaps of the cells. Make sure you are selecting the correct cells over time. You can adjust the selection and regenerate components even after you have already clicked Generate. When you reach the last time point, advancing one more time point brings you to the first one. You can remove the script for mask saving, since we don't want to make changes to the masks for now. Open the Tracking window and click Generate to automatically compute the association among the segmented cells. Once the calculation finishes, go to the Selection panel of the Tracking window and click Full Component to load all cells. To examine the tracking result, Selection_Tracking script. You may need to type all in the ID field and press Enter to refresh and load all segmented cells from the first time point. You can play back or step over time to examine the tracking result. Part 3. Automatic Tracking. We can further automate the component analysis process and use another 4D script to generate components for each time point. First, enable 4D script. And then experiment with the component analyzer settings to generate a good segmentation. The threshold value can influence the segmentation result. We would like to find a value that colors each cell correctly. Once a good segmentation is found, load a 4D script called generate_comp. Click the play button to execute the 4D script on each time point. You can observe the segmentation result at each time. You can go back to one time point and adjust the component analyzer settings to improve the result. When all time points are segmented, go to the tracking window and click Generate in the Track Map panel. After it generates the tracking of all cells, save the result. To examine the tracking result, load a Selection_Tracking script. Go back to the first time point. Select one cell and click Full Component to load for examination. Play back or step over time to see if it is tracked correctly. You can also select and load multiple cells. Part 4. Fixing tracking issues. There can be tracking issues when automatic methods are used. Use the paintbrush tool and manual tracking features to fix the issues. In this example, 2 cells have been identified as one component. We would like to assign a new ID to the smaller cell, and then link it to the correct cell in the previous time point. Erase the larger cell from the selection using the Unselect brush. Make sure that only the smaller cell is selected. Go to the Modify panel of the tracking window. Click Assign ID to assign a new ID to the selected cell. After fixing the segmentation issue, we may try to use the Refinement tool in the Track Map panel to see if it can be linked automatically. If the automatic refinement doesn't work, we need to link cells manually. Select the cell from the previous time point. Click Full Component to load the entire segmented cell in the previous time point. Click Exclusive Link in the Linkage panel to link the 2 components in the current and previous ID lists. Examine the tracking by stepping over time after fixing the issue. Now, the cells are tracked correctly. Go to the Track Map tab and click Save to save the modified result. Part 5. 4D Analysis. Load the 4D dataset and its tracking result generated previously. Enable and load a 4D script called component_analysis, which can be found in FluoRender's installation directory. Select one cell and click Full Component in the tracking window. Play back the time sequence and generate analysis information in the meantime. The analysis results are saved in a text file specified in the script. Open the file with Excel. Draw a plot of the cell's Z position over time. The cell's movement has a trend of going deeper. Draw another plot of the cell's mean intensity over time. The cell's mean intensity is decreasing for the last several time points.